Wow, it's that fated moment, the last video of the course, and we're going to address some finer aspects of vision here. Duplicity theory of visions is going to give us the reasons for why we have both rods and cones. We cannot have a receptor that can possibly be incredibly sensitive and yet also employ a very defined resolution to a processing signal. And so we're going to see one type is one we need for high sensitivity and the other type we need for high resolution. And so it makes sense to have one type of receptor for night vision and one type of receptor for day vision. Okay. So we need incredible sensitivity with rods. We need incredible resolution with cones. So rods need to be sensitive and react in the most dim light so that we can detect even the slightest ray of light. And so we're going to see this process called convergence occur, where you're going to have dozens of rods that converge on a single bipolar cell. And so if any one of those rods detects a ray of light, it will activate that bipolar cell, which will tell the ganglion cell, hey, we have light here, and it will cause the brain to realize that we have light reception. So in a typical situation, 600 rods will converge on a bipolar cell. Now, what level of complexity I still hadn't introduced yet is that many bipolar cells will converge on each ganglion cell. So now you're multiplying the sensitivity that we expect from the rods to detect very small amounts of light. So this gives us a really great sensitivity to light detection. Okay, but we can't expect resolution out of this. We can't expect by taking these big old fields of rods that we'll be able to say, oh, we can define something down to that level. No, it's going to be kind of fuzzy, kind of grainy is what we're going to expect to get. Now at the edge of the retina, you have rod cells that are widely spaced, and those are just for motion detection. You're not looking for resolution, or you're looking for sensitivity here, right? Because you want to know if something's coming out from the edges of your vision that you need to detect and react to. Okay, so once again, not worried about detecting exactly what it is, just that there is something there. So color vision is very different. We've got the fovea where there are no rods, and we have the highest concentration of cone cells that we can possibly expect inside the fovea. So obviously, because there are over 1 million cone cells, we have them spread throughout the retina. But in this place, this is the exclusive zone for cone cells. There's no convergence. You want resolution. You want the sharpest possible vision you can produce to occur here, and you want no convergence. So each cone cell has its own direct line to the brain. There'll be a single bipolar cell and a single ganglion cell, and those will take that information directly into the brain, especially expressly from the fovea here. That's why it's the center of the sharpest possible vision. And so by utilizing this type of process, we get incredibly high resolution vision. So there's less sensitivity, but incredible amounts of definition that we have here. So you can see this single channel effect where one cone acts on one bipolar, which acts on one ganglion cell. Whereas for the rods, you have massive fields of rod cells that are acting on these bipolar cells that themselves will converge onto one optic nerve. So in this particular case, we might have a few thousand rods that are going to eventually converge into this one ganglion cell. And result in the firing of one optic nerve fiber. Whereas here we can have multiple optic nerve fibers firing due to activation of many separate cone cells. So we talked about color vision. Here's our black and white vision right here, but we have optimized at blue, we have optimized at green, and we have cones that are optimized for red detection. All primates pretty much exhibit this type of system. But you might expect, like for cats and so forth, they would have mostly rods. Maybe not only rods, but mostly rods. And then the 3 types of cones are named, red, green, and blue, depending on what wavelength they are detecting. Okay, so the color perception is on your mix of how your rods are going to send this information to the brain. So color vision, hopefully you don't have color blindness. Hopefully you recognize that this color number in here is actually inverted and is not a normal oriented number. Okay, so it is possible to have a missing set of cones. You could miss completely the rod cones. You may also just have reduced sensitivity of these cones, so that, yeah, you still can detect some of the nuances of colors that are associated with red, but perhaps you can associate right at the red level. So there's also that occurs, and that's a little more common. To completely lack a color sensation is somewhat rare. To lack two of the color cones is really rare, and to lack all three is very, very rare. And so that would be complete color blindness, and you only see the world in grays. Again, very, very rare. The most common is red color blindness. The next most common is green, and then red-green. Blue is very rare, and red, green, and blue is incredibly rare. Okay, so it'd be difficult to detect shades. This occurs in many more males than females because of the way that it's transmitted via an X-linked gene. Okay, so when we talk about three-dimensional vision, we're talking about depth perception. This is the ability to judge depth. And so what we require is overlapping visual fields. This is going to help us determine depth, and let's see how that works. So the bipolar cells will fire into the ganglion cells, and then the optic nerves, or the nerves from those ganglion cells, will send back axons to form the optic nerve. So we have the right and the left optic nerves, and then they're going to converge into the optic chiasm. Aha, what happens there? Well, half the fibers from one side of one optic nerve will stay on the same side, the other half will cross over. Half the optic nerves from this eye will cross over, and half will stay on the same side. And so they split to form the optic tracts. And so the right cerebral hemisphere only sees objects in the left visual field. We'll describe exactly where those are in just a second. And the left cerebral hemisphere will see objects on the right visual field. Okay, and it makes sense that we're still associating that with the right half of the brain controls the left half of the body, the left half of the brain controls the right half of the body for the most part. So let's look at how this works. Also, we need to mention how the tracts go back. So once we form the optic tract, it's going to go to the thalamus, because remember, all sensory input goes to the thalamus, and the vision is no different. Only smell does not go to the thalamus first. And so it will go to a region of the brain called the lateral geniculate nucleus. And then from the lateral geniculate nucleus of the thalamus, we'll have these huge optic radiations that will go back to the occipital lobe. Okay. And in the occipital lobe, we'll invert the images, fix them, make them in the correct orientation, and do all the color mapping we need to do, all the light detection that we need to perform here. All right. Now, don't forget that our superior colliculus also receives just a few of these visual fibers to aid us in bright flashes, and so that we can respond appropriately with our startle reflex. So here's the idea of forming a visual projection pathway, and you can see how some of the fibers don't cross over, some do. And let's look at this in a little more detail here in this graph, which I think shows it a little better. So in the optic chiasm right here, we're going to see that in the left visual field is formed from what's called the temporal side of the right eye and the nasal side of the left eye. So these fibers form the left visual field, and you'll see this nasal portion of the left eye, the fibers that come from it will decussate in the optic chiasm, but the fibers from the temporal portion of the right eye will not decussate, they'll stay on the same side, so that this left visual field will be processed by the right side of the brain. Now on the left eye, for the right visual field, for the left eye, we'll use the temporal portion of the left eye's retina and the nasal portion of the right eye's retina, and you'll see the nasal portion of the right eye will cross over and decussate, but the temporal portion will stay on the same side. Okay, so that's great, right? Makes sense. And so that will form, we'll have the big optic radiation, so the right visual field will have the left side of the brain act on it. Don't forget, there's a big stopping point here, and that's the lateral geniculate nucleus of the thalamus. Going back to the primary visual cortex is where we'll do most of the visual processing, but also understand a large portion of the cerebral cortex is devoted to visual processing. At any one time, 30% of it is devoted to processing visual information, quite a large proportion. So this just shows you the same aspects of fiber crossing over, these huge fibers, these huge sets of axons that go back to the left and right cerebral hemispheres that are called optic radiations. So we know that some visual processing begins in the retina, where we have some adjustments for different aspects of the visual information, but then it's the primary visual cortex that's going to receive the data and send that data forward to be processed in other regions of the brain, right? And lots of things are going to be detected here. So here we see those massive optic radiations that are coming out of the thalamus, projecting back to the occipital lobes of the respective hemispheres. That's the one beautiful shot I love in this picture. Shows you a beautiful pathway where you have the optic nerve, optic chiasm, optic tract, LGN to the radiations back to the occipital lobe. And this is shown to you in highlight here in illustration purposes, and each side's fibers are demonstrated in these other figures. I want to thank you for what's been a tremendous learning experience for me. This is the last visual video for the semester. You still have your one, though, for review for exam 3. Wish you all the best of luck in the future. If I see you again for the second half of this course, for the next online portion, it will be the first time I'll have presented that one as well. I'll welcome you with open arms. I appreciate any input you've got to give me, and I appreciate all the support and help I've got. You guys have been more than nice in your presentation to me, in your discussion of what I could do better, and I've been very patient, and I truly, truly appreciate that. Best of luck to you.